Horticulture contributes billions of dollars to the world economy in fresh produce, processing, employment opportunities, in plant and equipment, and through associated industries. But the health of those industries and the livelihoods of people who work in them is under threat from a disease called crown gall. Crown gall attacks woody plants like fruit and nut trees, some nursery species, and roses. The problem is caused by a bacterium called Agrobacterium tumefaciens, which occur naturally in the soil. When plant material is injured, soft tissue becomes vulnerable to attack from the bacterium.
Bacteria in the soil invade the roots of the plant and tell the roots to divide uncontrollably and cause an outgrowth tumor, as you can see here in this plant, which makes the plant unhealthy because it can't uptake water and nutrients as effectively.
There is no known treatment for a plant infected with crown gall. The disease had a terrible impact on the viability of horticultural industries.
Some years it varied from perhaps a 5% to anything up to 50% rejection, or bonfires with half the trees had to be destroyed because of the crown gall problem. It's very, very severe.
They don't grow as good, and the fruit size is a lot smaller, and the trees, of course, it's not very healthy.
And the other problem you had too, of you had all these orders that you hopefully could fulfil, and then you had no idea really of what percentage you were going to throw out. So what you thought you might have had 50,000 peach trees suddenly turned out to be 25. You're in big trouble.
With no way to cure a plant infected with galls, researchers decided the best way to deal with crown gall was to prevent it. In the late 1960s, Alan Kerr, an Australian research scientist, went to an orchard near Adelaide, South Australia, in search of a biological control for crown gall. There exists in nature a natural enemy of almost every pest and disease. Alan Kerr's challenge was to find the bacterium which caused crown gall, then find its natural enemy. It was arduous work.
Well, I suppose the first problem was that we had to distinguish Agrobacterium in the soil from 100 million other bacterial cells per gram of soil.
Professor Kerr's team took samples from the Balhanna orchard. This patch of ground, still part of a modern-day fruit nursery, yielded the breakthrough result.
We found that there were two different kinds of bacteria, one that caused crown gall and one that didn't cause crown gall. And when we mixed these two together, we found that the one that didn't cause crown gall prevented crown gall being developed from the first bacteria.
In the soil where Agrobacterium tumefaciens, or the bad bacteria, are found, there was also a good bacterium. It is called Agrobacterium radiobacter and codenamed K84. The good and bad bacteria are cousins. They're very similar. The good bacterium, or K84, produces a natural antibiotic called agrocin 84, which protects the plants from the bad bacteria. When K84 is applied to plant material, it produces the natural antibiotic agrocin 84. The antibiotic essentially forms a protective layer, a shield, around injured plant tissue. When it is in place, the bad bacterium can't get through to exposed plant tissue. K84 was incorporated into a water solution so plants can be dipped and treated with the biocontrol agent. K84 was exactly what the horticultural industry needed. Simply by dipping plant material in K84, producers prevented the devastating disease crown gall.
When Dr. Alan Kerr came up with his little trowels and plastic bags and took samples off our original nurseries blocks back at Balhanna, and we used to think, well, what the hell is he doing there, and used to laugh about it all and wonder whether anything would come of it, but as it turned out, it was one of the best things that ever happened to the fruit growing industry and the nursery industry.
K84 virtually eliminated crown gall disease. Then disaster struck. The bad bacteria were becoming immune to K84. When good and bad bacteria are in close proximity in the soil, the bad bacteria are able to steal the antibiotic-producing plasmid of DNA from the good bacteria. Once in possession of this genetic material, the bad bacteria become immune to the antibiotic agrocin 84 agent produced by K84. With this breakdown in the antibiotic shield, the bad bacteria are once again in a position to produce cancerous galls and choke healthy plants. Professor Larry Moore of Oregon State University has studied the transfer of genetic material from one bacterium to another.
Experimentally, we've demonstrated that Agrobacterium tumefaciens, the bad guy, has received plasma DNA from the good guy, K84. This was done in the field. And when we recovered isolates of Agrobacterium from the tumors that developed on the cherry plants that were inoculated, these isolates, at least some of them, did indeed have characteristics of both the bad guy and the good guy.
Although immunity threatened previous work on the biocontrol of crown gall, science again came to the rescue with research which allowed plant biologists to remove from the good bacteria the very piece of DNA which allowed immunity to occur.
The resistance developed because something was being transferred from the control organism to the disease organism. And all that we did was to stop that transfer, so that we located the genes that were involved in the transfer process. We cut them out using genetic engineering and then put them back into the original cell again.
Apart from the deletion of a tiny piece of genetic material, the good bacterium, codenamed K1026, remained exactly the same as K84. K1026 was still able to produce the antibiotic shield of agrocin 84, which was effective against gall-forming bacteria. K1026 was incorporated into a product called Nogall. Nogall produced the same good results for growers and producers in the horticultural industry.
It's as effective as the other product. It's used the same way, and growers can now relax because the issue of resistance in the field is eliminated.
Nogall is produced under licence by BioCare Technology just outside Sydney, Australia. BioCare is licensed to produce and market Nogall worldwide by Luminous Proprietary Limited of the University of Adelaide. An enormous amount of scientific and technological research goes into not only finding biological enemies of pests and disease, but also making those biocontrols into effective and inexpensive commercial products. Biological controls and genetically manipulated organisms are subject to the strictest scientific scrutiny to make sure they have no negative impact on the environment or humans. Nogall represented a number of technological breakthroughs.
The development of this new product is significant because it continues and enhances a landmark biocontrol agent as far as the history of plant pathology is concerned. Crown gall has been certainly, the risk and the threat of crown gall has been greatly reduced all over the world where susceptible plants are grown. That's major. The science behind this has also been impressive. The identifying of the genes involved as far as the mechanism by which the biological control agent works. We have a good handle on that. Not all the answers, but a good handle. And because of that, they were able to genetically engineer the strain to reduce the potential for Transfer of the agrocin plasma to Agrobacterium tumefaciens, a pathogen, and thus help to ensure the longevity of use of this control agent. So both practically and scientifically there have been some very strong pluses to both the profession and science and to agriculture in general.
And in a world where there's increasing concern about chemicals and food, biological controls offer a clean, green, natural solution.
The potential for biological control in many industries is enormous. The benefits to the industry are that they have less chemical, less residues, a safer, cleaner product at the end of the day, and it's sustainable.
But perhaps the best thing about the Nogall success story is its impact on the industries it was developed for.
Since we've been using Nogall, we've been getting better results because the trees, when they're ready to pick fruit, they all come at the same time. And if you don't use Nogall, picking season extends longer and you get a lot more waste in the fruit.
It's so simple to use. I mean, it's just a matter of tipping a packet into some water and dipping the seeds or the rootstock into it. No smells, no mess, no nothing. It's just brilliant.
The report on it is very good. Our own experience in Oregon has been that this strain compared to the old product proved equal to it in preventing crown gall on a variety of woody plants that we had planted to the field. I'm aware of some excellent work in Spain where it also was very efficacious, equal to K84 in all respects, or the old product, and in one instance actually showed an improved activity against a strain that was resistant to the old product.
The development of Nogall is significant for several reasons. It is founded on a solid base of scientific research, enhanced by innovative genetic manipulation, and married to commercial technology, which makes it accessible to its end users. This in a product with proven efficacy against crown gall in the species it targets: stone fruit and nut trees, some nursery species, and roses. Nogall for effective control of crown gall.